Hello everyone. Today we're going to talk about why creationist kinds definitely don't exist. So let's jump right in. All major anti-evolution organizations accept the process of speciation, and as we saw in the video, do dogs produce non-dogs, evolution is a process of repeated speciation events. In sexually reproducing organisms, populations that begin to diverge into separate species are at first usually only distinguishable by very minor genetic and morphological differences. Take, for instance, the marbled crawfish, Procambarus virginalis, which was discovered in the aquarium hobby in 1995. This species is descended from P. phallax, but its lineage experienced a mutation that changed it from diploid to triploid. Morphological differences between P. virginalis and P. phallax are pretty minor, such as P. virginalis having larger maximum body and clutch sizes on average. Additionally, the two are so similar morphologically that they still attempt to mate with each other. However, this fails because P. virginalis can only reproduce clonally. As humans have been aware that speciation can, and does, occur for only about a century and a half, the number of cases of observed speciation events are rather few. Some experiments have also shed light on this area, such as one with Drosophila pseudoobscura. In a 1989 experiment, researchers divided 8 homogeneous populations of these flies into 2 groups of 4, feeding one group maltose and the other starch. Within one generation, the maltose and starch flies became reproductively isolated. This event was due to their gut flora, in particular Lactobacillus plantarum, producing volatile metabolites that affected the fly's ability to identify suitable mates. Of course, most creationists don't have a problem with examples such as these, at least not anymore. Creationists used to deny the possibility of speciation, but they have now moved the goalpost a bit. Currently, when they are presented with, for example, new species of fruit fly or fish, they will often scoff and dismiss them by saying, "But they are still fruit flies," or "They are still fish, they are still the same kind, they didn't turn into something else." Or more extremely, creationists dismiss evolution experiments with bacteria on the grounds that these bacteria are still bacteria. For example, during the abysmal God vs. Evolution movie, if you can call it a movie, Ray Comfort asked for examples of Darwinian evolution. P.Z. Myers provided several examples, such as Richard Lenski's long-term evolution experiments on E. coli. Comfort simply stated that they're still bacteria, which is rather silly. Bacteria are extremely diverse in their biochemical properties. Some can photosynthesize like plants, others can obtain energy from organic compounds with oxygen like animals and fungi, but others can obtain energy through a host of means. Some can even use inorganic chemicals like hydrogen, iron, or sulfur. The diversity and disparity of bacteria make elephants and pine trees look like sisters by comparison. The fact is that every organism is always part of a larger group or clade, which in turn is part of a bigger clade, and again and again. Both humans and chimpanzees are still hominids. They and monkeys are still primates. Primates and marsupials are still mammals. Mammals and reptiles are still vertebrates. Vertebrates are still animals, and all animals are still eukaryotes, as are plants and fungi. So the "they are still" insert group response works equally well at every level of classification, no matter how far you go down the rabbit hole that creationists have to deal with for determining their kinds. This argument also sneaks in a gross straw man against evolution, as it implies that from evolution we would expect organisms to evolve in a way such that they are no longer part of the group from which they originated. This is indeed false, which we also addressed at length in the Do Dogs Produce Non-Dogs video. Speciation producing similar yet distinct species which remain members of their parent groups is exactly what we always expect from evolution. Creationists could make the argument that speciation only generates slight differences, so it can only produce sets of species with slight differences between each other, such as cats. It cannot lead to species with large differences between them, like all mammals. So, in their mind, evolution would need to make impossibly huge steps to bridge the gap between distantly related species. This mindset would explain the common misconception of what transitional species and common ancestors would have looked like. To bring up Ray Comfort again, he and Kirk Cameron are infamous for their crocoduck, thinking that the common ancestor of crocodiles and ducks was some direct hybrid between them. However, this is also a straw man of evolution. Even Darwin explained why we don't expect such absurd blends. Distantly related species aren't separated by only one speciation event. Of course, the first speciation event did cause the ancestral line to split from their common ancestor. However, from that point onward, multiple speciation events would take place in both lineages, each generating slight differences, which accumulate over long time periods. The unique traits that we see in ducks and crocodiles respectively were derived during the time after they diverged from their common ancestor, so the common ancestor itself would not have possessed these traits. This is why we would not predict to see a crocoduck. But what do we predict? Well, according to evolution, the big differences between clades of organisms are the product of slight differences that were added up over long periods of time. If this is true, then when we look at these clades and track them backwards through time, we should observe species of these respective clades that would begin to look more and more like each other the further back you go, until they are barely distinguishable. Specifically, when you come close to the initial speciation event. At that time, there were only slight differences between the lineages that only recently speciated, which is how every clade started out. Fortunately, there is a way we can test our speciation prediction over long timescales: the fossil record. Where this applies very well is to lineages with exquisite fossil records, such as foraminifera and diatoms. As paleontologist Donald Prothero notes in Evolution, What the Fossils Say and Why It Matters, quote, for example, in the Pliocene, one of the common planktonic forams is Globigerinoides sacculifer, which has a shell shaped like a series of porous oblong bubbles clustered together in a spiral arrangement. Through the many cores that sample the Pliocene oceans, we can find more and more specimens that develop these long, slender, finger-like extensions all over the final few chambers. As you move up the cores, these little fingers become longer and more common. These creatures are so distinct from the ancestral lineage that they branched away from that they are given their own species, Globigerinoides fistulosus. They do indeed look like little fists. Close quote. But this prediction also works even when a lineage's fossil record isn't so well preserved. Take humans and chimps, for example. Today it seems like there are a lot of differences between them. Humans have invented technology, our capacity for language is far greater, we walk obligately bipedally, our hair isn't as thick, the proportions of our teeth are somewhat different, and our face is not nearly as prognathic as that of chimps. However, these differences are not all simultaneously present in our earliest hominin relatives, like Sahelanthropus. And they are not even all present in members of our own genus. Just look at Homo Habilis. As a 2005 paper describes Sahelanthropus, quote, in terms of cranial vault shape, Sahelanthropus resembles P. troglodytes and G. gorilla in having a low, long, superior contour of the neurocranium, particularly anterior to the apex of the vault. The nuchal plane in Sahelanthropus, however, is relatively long, flat, and rotated toward the horizontal, as in A. africanus and A. afarensis. Relative facial height from Nasian to Prosthean in Sahelanthropus is similar to that of A. afarensis and A. africanus and H. habilis, but shorter than that in G. gorilla and P. troglodytes. Close quote. In other words, slight morphological differences in the cranium distinguish early members of our lineage from the chimp and gorilla lineages. That's not nearly as dramatic. Pushing back to the common ancestor of platyrrhines and catarrhines, the late Eocene Proteopithecus is a stem anthropoid whose dentition is transitional between parapithecids and crown anthropoids. According to a 1999 paper, quote, Proteopithecus sylviae shares with parapithecids, e.g. Apidium and Parapithecus, a few features that are likely to be primitive for the suborder, such as having 3 premolars and extremely broad cheek teeth, with P2 smaller than the other premolars and M3 smaller than the other molars, close quote. Also, quote, P. sylviae resembles many living platyrrhines, particularly in having a relatively large upper P2 and a lower P2 that is smaller than the other premolars, and also because the upper molars of platyrrhines tend to be broad. Among platyrrhines, P. sylviae shares more features in common with Saimiri, Sanguinus, and Calamico than other New World monkeys, especially in having a well-demarcated trigon and a well-defined hypocone, close quote. Further, Proteopithecus is similar in its dentition to the early platyrrhines Brennusella, Dolichocebus, Soriacebus, and Carlocebus, indicating that a Proteopithecus-like ancestor may have rafted from Africa to South America and founded the platyrrhine lineage. Another African anthropoid, Talapithecus, is also strikingly similar in its dentition to the early platyrrhine Perupithecus. A 2015 paper notes, quote, Talapithecus is similar to Perupithecus in size and overall morphology, and differs only in some minor details, such as having the buccal side basal to the metacone slightly developed, close quote. As a result, Talapithecus and Perupithecus have been placed very close to each other phylogenetically, despite the former being from Africa and the latter being from South America. This biogeographic pattern is likely the result of rafting from Africa to South America, as we explained in the video, A Critique of Intelligent Design Part 1. So again, despite platyrrhines and catarrhines being quite different today, their split was originally due to minor dental and postcranial differences. The same is true of the earliest perissodactyls, the odd-toed ungulates, rhinos, horses, and tapirs. Prothero writes, quote, the specimens of the earliest horses, now called Protorohippus, and the earliest tapiroids, Homo Habilis, were virtually identical, except that the Homo Habilis molars had slightly better developed cross crests, a signature of the teeth of all later tapiroids, close quote. And, quote, the earliest rhino relatives like Hyraceus are barely distinguishable from contemporary tapiroids in the early Middle Eocene, close quote. So, are rhinos, tapirs, and horses all one kind or independently descended from nearly identical ancestors? Sister to the perissodactyls is a family known as Phenacodontidae, which are generalist herbivores, and based on molecular data, the closest relatives of hoofed mammals are pangolins and carnivorans. This relationship is further supported by a host of Paleocene generalist omnivores, such as the families Arctocyonidae and Hyopsodontidae, who may be representative of the ancestors of hoofed mammals. One known as Creacus was re-examined in a 2020 paper that says, quote, the inner ear of Creacus also shares derived characteristics of the elliptical and spherical recesses with extinct species that belong to Ungulata, the extant placental group that includes artiodactyls and perissodactyls, close quote. Again, in what kind do these mammals fit? Let's switch to the carnivorans now. All extant members are in one of two clades, Caeniformes, dogs and their kin, or Feliformes, cats and their kin. Dogs are members of the family Caenidae and all run on their toes. They are digitigrade. This was not always the case, though, as the earliest canids were instead plantigrade. They ran on the soles of their feet. This is represented by such canids as Hesperocyon. As a 1993 paper writes, quote, compared to the archaic Miacoid carnivorans, Hesperocyon has a number of advanced features suggestive of running adaptations, such as absence of a large teres major process on the scapula, a relatively reduced deltoid crest on the humerus, elongated and adjoined metapodials, and laterally oriented fovea capitis femoris. On the other hand, Hesperocyon retains some primitive arboreal characteristics, such as a short, broad scapula, with long acromion, a stout humerus with a deep bicipital groove, and a thin-bladed brachioradialis crest, an interiorly bowed ulna, a sharply angled pelvic floor, a relatively large muscle scarf or flexor digitorum longus and caudal tibialis on the tibia, and a short, deep, and hooked distal phalanx suggestive of a retractile claw, close quote. Basally derived with respect to carnivora are Miacidae and Viverridae. Miacidae is actually paraphyletic to carnivora, with some members being more carnivoran-like than others. For example, Vulpavus is more carnivoran-like than Dormalicyon, which is more carnivoran-like than Gracilocyon. Quite a nice gradual evolutionary sequence, don't you think? That is, unless one wants to argue that separate, slightly more carnivoran-like kinds were created, simply coincidentally mimicking an evolutionary relationship. As miacids form the base of carnivora, they are the ancestors of both dogs and cats. In fact, Raw Matt from the Standing for Truth Brain Trust made a video wherein he argued that the original cat ancestors were miacids, but they were quick to quietly delete this from their channel, likely after they realized this would have some very awkward implications to young Earth creationism. Oh well. And the last two clades we will look at are Proboscidea, the elephants, and Sirenia, the manatees and dugong. While clearly different today, the earliest elephants, such as Meratherium, and the earliest Sirenians, such as Pesosiren, look oddly similar to each other, which makes sense to people who accept evolution because the two are sisters genetically. Why then are they so different today? A 2010 paper explains. Quote, when developing biogeographic scenarios, it is important to consider locomotion. The partial innominate from South Carolina, like many other early Sirenian innominates, appears at most to be barely capable of bearing the animal's body weight on land. Even Pesosiren portelli, whose innominate appears to have been adequately supported to have allowed terrestrial locomotion, had pachyosteosclerotic ribs, indicating that it regularly fed on underwater plants. But if aquatic plant herbivory is found in all of these early Sirenians, why is there such similarity between the innominates of Protosiren and the Middle Eocene Proboscidean Meratherium? Basal proboscideans are clearly in a different clade from early Sirenians, but the similarity in their pelvic girdles raises interesting questions. The innominates of Protosiren are quite similar in their morphology, including sacroiliac joints, obturator foramen size, and rami thicknesses, to the innominate of Meratherium from Egypt. Meratherium did not have pachyosteosclerotic bones or a downwardly deflected rostrum like Pesosiren, but some recent studies indicate that it may have fed on freshwater aquatic vegetation. The depositional environment and stable isotopic studies of Eocene Sirenia from Egypt, including Eosiren and Protosiren smithae, indicate that these animals were already feeding on seagrasses. So despite the potential similarities in body form, including their innominates, it appears that Meratherium and Protosiren led lives very different from each other. But if Meratherium and Protosiren had similar terrestrial locomotor capabilities, why wouldn't Meratherium, or any other early proboscidean, have been found in North America? The obvious reason is already known: their ecology. The stable isotope record indicates that they differed in their use of freshwater versus marine environments, and this would have affected their potential distribution. Perhaps the freshwater feeding of Merathers limited their distribution, and the diet of marine seagrasses led Protosiren to both sides of the Atlantic. Close quote. Again, subtle differences in diet led to particular biogeographic distributions, putting the two orders down separate evolutionary paths. In all of these cases, whether we are looking at recent speciation events or fossil trends in deep time, we can see that a series of small, natural changes accumulate over successive generations, ultimately resulting in dramatically different organisms. This is how evolution works. This is how mutations, natural selection, and the other mechanisms of evolution have generated the vast biodiversity that exists on Earth. Here I mostly covered examples from mammals, but if you are interested in seeing transitional forms among, say, dinosaurs, then we covered them in the Dinosaur Phylogeny series. So, thanks for watching, and I'll see you next time.